Thank you. My name is Martha Volkoff. I'm the Environmental Program Manager for California Department of Fish and Wildlife's Invasive Species Program. And our program covers aquatic invasive species and any other, I guess, invasive species that arise. And our program has had a quagga zebra mussel program. We have quagga and zebra mussels in the state of California, and those populations were in Southern California, but this is a new species, golden mussel, that I'm here to share with you today. Next slide, please. So a little bit, if you're unfamiliar with invasive mussels, because thankfully at this point, the Pacific Northwest is free of populations of any of the species, the golden mussel, again, which will be the focus of today. But I also wanted to, given that you may not deal with these species, a little bit of overview about invasive mussels collectively, including quagga mussels and zebra mussels, two other species that are a threat to the Pacific Northwest in addition to golden mussel. So all three of these, for the moment, I'll talk collectively about all three of these species. They're fresh and brackish water bivalves, meaning they have two shells. They have byssal threads, and in the illustration at the bottom, circled in the red circle, is the byssal threads, which allows them to attach to hard and semi-hard surfaces, meaning they can attach to man-made surfaces as well as natural surfaces. These are small mussels, not exceeding 2 inches in length. However, they do form dense colonies, and they're individually they're small, but collectively their impacts are magnified over those dense populations. And they can vary in coloration now across these three species, a little more so than when we were just dealing with quagga and zebra mussels that usually were striped. Golden mussels have a more uniform coloration. And something that is shared across these species, and that is the attribution for why they're invasive and a threat to our environments, is that they tend to have a wide ability for different environments, including water temperatures, salinities, nutrients, disturbance, and and I mentioned their dense colonies. These dense colonies of mussels compete for other, I blocked out phytoplankton. So they can strip out, again, these mass populations of these mussels if they form in really suitable environments that allow these large colonies, can strip out the phytoplankton in environments and other particulate matter, resulting in increased water clarity, which in some cases might be a positive thing. In other cases, it has other consequences, including increased water clarity and aquatic plant growth. They form those dense colonies on surfaces requiring, impacting those surfaces, maybe requiring maintenance and removal in an ongoing long-term situation, changes in water quality, and at the crux of it, altering aquatic ecosystems. And today I'm focusing on environmental impacts. There's a whole nother conversation about infrastructure impacts. I'll talk a little bit about that relative to California's water conveyance systems for your extrapolation to your environments up north. Next, please. So I mentioned their population size, density, as well as I didn't talk about their fast growth rates in suitable environments. So in California, we have one population of zebra mussels in a small reservoir in Central California. And there was some early work in 2008 when those mussels were established or detected, monitoring the population growth in that reservoir. And the next 3 slides, is a time series over 23 weeks of their dense colonization and rapid growth. So if you could just slowly click through the next 3 slides, we see at 8 weeks, 12 weeks, this is the same plate that was submerged in the lake. And this is an example of the biofouling and the dense populations that can result. These are zebra mussels, but this is also true for quagga and also golden mussels. The photo of the shoreline on the larger picture is that same reservoir, San Justo, and you see on the rocks above and below the water line. So the water was receding in this photo, exposing those rocks that were once submerged, of completely coated with these zebra mussels. And then the mussel on the left, this photo is not from California, but pulled from elsewhere in quagga and zebra mussel range, because it's an earlier photo. But when there are hard substrates, can include native mussels. And so this dense colonization on other mussel species can impair that substrate mussel from its filter feeding and survival. And so there is concern for native mussels being colonized by golden mussels and quagga and zebra mussels. In addition, the photo on the right is a propeller from a watercraft. So any things that are submerged in these invaded environments is also subject to colonization by the golden mussel and invasive mussel, speaking generally again, impairing that equipment for its operational use and added maintenance. Next slide. A little bit about the life cycle of all of these mussels is important to understanding their threat of introduction, as well as some management opportunities. So the lifespan is variable, probably across different environments, but typically these mussels survive 2 to 4 years. They reach spawning size at a fairly early age, post-settlement. They release their eggs and sperm into the environment, again, driven by water temperatures. And so the environment they're in can be controlling for that, essentially, in terms of it may prohibit them from establishing an environment if the water temperatures aren't high enough to initiate spawning. And the length of time those villagers and eggs take to develop to settlement stage varies. So they release the eggs and sperm into the water. They find each other, or if they find each other, fertilize those eggs, and those eggs develop over a couple of weeks, again, water temperature dependent, to go through their different stages of development, until they reach the stage at which they settle, and they attach to a surface, hard or semi-hard, form those byssal threads. And so that's one key for identifying non-native mussels and differentiating invasive mussels from other species in the environment. If you touch them and sort of wiggle them, they're attached. you can knock them off, but more or less they're not going to slide like some other species may, and none of the clam species, which would be in the substrate, you wouldn't find clams attached like you would the invasive mussels. Not to say these mussels don't fall off and end up in the substrate, so not entirely going to differentiate, but if you do find a bivalve attached to a surface in a freshwater environment, that is a very indicative of a non-native species that is cause for concern. Okay, I think that's all I have to say on that. Oh, so this veliger life stage is subject to being moved and transported overland in entrapped water. And another important factor of these species is that they can survive either in entrapped water or the larval veliger stage. So think in the ballast of boats or in buckets of water or any other standing water in watercraft. Also, the adults, as that photo of the prop of the boat showed in the prior slide, the adults can attach to watercraft or other equipment that is removed from the water. Their shells close up tightly, allowing them to withstand being out of the aquatic or out of water and attached to surfaces for days up to weeks if it's a suitable temperature and humidity. So they're very resilient. And that is one of the reasons that they are able to move across through human-mediated pathways over land. Next slide, please. In California, just to give you a geographic range of where golden mussels have been detected, this is the first detection of golden mussels in North America, and it is in the Sacramento-San Joaquin Delta, which is in the red box on the map. We believe they were introduced by a transoceanic ship that came into the Stockton port, which is a freshwater port. All of these waters are highly interconnected. The delta is highly modified, highly invaded by other species, and is a mixed use environment. Essentially, it is an important habitat for native species. It is also highly developed historically for agriculture. The islands in the delta, well, we've created islands in the delta by building levees. It's an important agricultural area, and as California continues to increase in population density and increasingly urbanized and developed human area. These golden mussels are native to China and Southeast Asia, but other areas that have been invaded include Hong Kong, Japan, and South America. And for California, we consider nearly all of California's fresh and brackish waters are suitable for golden mussel establishment. We had some buffer for quagga and zebra based on their calcium requirements, but golden mussels can survive and reproduce in significantly lower calcium waters than quagga and zebra mussels. Next slide, please. This is just zooming in on that red box on the prior slide. We have, with the detection of golden mussels in October of 2024, in the fall, we did a pretty rapid assessment within the delta to determine the extent of the infestation. This map is from, I believe, back in February 2025. And so all the dots may not be within the delta, but for this purpose, it works today. The Delta is an important hub for water conveyance to points south in California. So the blue and black line in this map indicate the state and federal water projects that pump water out of the Delta and send it to the southern San Joaquin Valley, or in the state water project down to Southern California through a complex network of canal systems, both above and below ground at different points through the system, as well as many pumping plants. So there is a significant impact of golden mussel detection in the Delta. At this point, there have been detections up to, I believe it's a couple 100 miles south of the Delta in that canal and pumping system. And we also have information from eDNA, older archived eDNA samples that they've been in the Delta back to spring of 2023. Next slide, please.
Martha, just about 2 minutes left.
Oh boy. Okay, skip through this slide.
Okay.
Basically, golden mussels can develop in or spawn in waters, at least based on the literature, down to 61 degrees. But again, these are very adaptable species, so I wouldn't take that as a limiting at this point. Some of the containment challenges include that water conveyance system, as well as the interconnected waters of the Delta and many unmanaged access points in the Delta. So that's a threat for overland spread, as well as the commercial shipping, again, in that port system. Next slide, please. So California has formed a task force. We have updated some laws that prohibit possession and movement of mussels. However, containment of exiting watercraft and equipment from the Delta is unmanaged, so there continues to be a threat to other states for the movement of golden mussels, water, and attached adults out of the Delta. And we are focusing on, within the funding and authorities we have, to continue to monitor throughout the state, conduct outreach and education, and partner engagement. Next slide. And my recommendations to others throughout the West and really across the US is to promote the clean, drain, dry messaging for watercraft and equipment every time someone launches, and also to implement watercraft inspections pre-launch to protect uninfested waters from invasive mussels and all aquatic invasive species introductions. And if you're not monitoring in your state, I encourage you to take that up, either using existing surface surveys or artificial substrates. And if you have greater ability to invest in other forms like plankton monitoring or eDNA sampling, that would also be good. Next slide. And then throughout monitoring, we did find another species of non-native mussels, Xenostrobus securus, in Southern California, coastal Southern California. Again, if you have the opportunity to monitor in more coastal environments, this is another brackish water species new to North America. I encourage you to do so. Next slide. I end right on time.